Altered cellular and tissue biology. Cellular adaptation is the process by which cells respond to both normal physiological demands and adverse pathological conditions. These changes are typically reversible and allow cells to maintain function under stress. However, adaptations to pathological conditions are often only temporarily successful. The major types of adaptive changes include atrophy, which is a decrease in cell size, hypertrophy, an increase in cell size, hyperplasia, an increase in the number of cells, dysplasia, characterized by abnormal cell growth, and metaplasia, where one mature cell type is replaced by another. This diagram shows the various forms of cellular adaptation. In a normal state, cells are organized and uniform, maintaining healthy structure and function. With atrophy, cells shrink in size, often in response to reduced demand or nutrient supply. Hypertrophy is the opposite, where cells grow larger to meet increased functional demands. Hyperplasia involves an increase in the number of cells, leading to tissue expansion. In metaplasia, one mature cell type is replaced by another, often in response to chronic irritation. Dysplasia represents disorganized, abnormal cell growth, which can be a precursor to malignancy if the stimulus persists. Atrophy refers to a decrease in cell size, which can lead to a reduction in the size of an organ if enough cells are affected. It can be physiologic, occurring naturally during early development as certain structures diminish. It can also be pathologic, resulting from decreased workload, reduced pressure, limited use, compromised blood supply, poor nutrition, or loss of hormonal or neural stimulation. Regardless of the cause, atrophy is a survival mechanism that allows cells to adapt to a less favorable environment by conserving resources. Hypertrophy is the increase in cell size, which leads to an overall enlargement of the affected organ. Physiologic hypertrophy occurs in response to increased demand or stimulation by hormones and growth factors, such as in skeletal muscle during exercise. Pathologic hypertrophy, on the other hand, often results from chronic hemodynamic overload, like the stress placed on the heart during hypertension. In both cases, the cells adapt by growing larger to meet the new functional demands placed upon them. Hyperplasia refers to an increase in the number of cells, driven by an accelerated rate of cellular division. Physiologic hyperplasia can be compensatory, allowing organs to regenerate after injury, or hormonal, occurring in organs that respond to specific hormonal signals, such as the uterus during pregnancy. Pathologic hyperplasia usually results from abnormal hormonal stimulation, leading to the excessive proliferation of otherwise normal cells. While hyperplasia itself is a controlled process, persistent stimulation can increase the risk for developing cancer. Dysplasia describes abnormal changes in the size, shape, and organization of mature cells. Although the tissue may appear disordered under the microscope, dysplasia itself is not considered cancer. Importantly, if the triggering stimulus is removed early, dysplastic changes may be reversible. However, if these changes penetrate the basement membrane, the process becomes an invasive neoplasm, marking a significant step toward malignancy. Metaplasia is the reversible replacement of one mature cell type by another, typically as an adaptive response to tissue damage, chronic irritation, or inflammation. It often occurs during repair and regeneration processes, helping the tissue survive under new conditions. This change happens through the reprogramming of stem cells or undifferentiated mesenchymal cells. Although metaplasia can protect tissues temporarily, persistent stress may increase the risk of progressing to dysplasia and ultimately malignancy. This illustration shows how chronic injury or irritation in the bronchial epithelium can lead to cellular adaptation. In response to persistent stress, the normal ciliated epithelium can undergo metaplasia, where it is replaced by a different type of cell better suited to withstand the injury. If the damaging stimulus continues or worsens, metaplasia can progress to dysplasia, characterized by disorganized, abnormal cell growth. Although metaplasia is reversible, prolonged dysplasia increases the risk of developing invasive cancer. Cellular injury occurs when a cell is unable to maintain homeostasis in the face of external or internal stress. If the injury is mild or short-term, the damage may be reversible, allowing the cell to recover and return to normal function. However, if the injury is severe, prolonged, or overwhelming, it becomes irreversible, leading to cell death. The ability of a cell to adapt or succumb depends largely on the nature of the stress and the cell's capacity for repair. This flowchart illustrates the cellular response to stress. A normal cell exposed to stress may successfully adapt, maintaining its function. If the cell cannot adapt, it experiences injury. When the injury is mild and temporary, the damage is reversible and the cell can recover. However, if the injury is severe or prolonged, it becomes irreversible, leading ultimately to cell death. This process highlights the delicate balance between cellular resilience and vulnerability under stress. Hypoxic injury is the single most common cause of cellular injury. It occurs when cells are deprived of adequate oxygen, disrupting normal cellular metabolism. Hypoxia can result from several factors, including ischemia, which is a reduced blood supply, reduced oxygen content in the air, loss of hemoglobin, or decreased production of red blood cells. It can also arise from diseases affecting the respiratory or cardiovascular systems, or poisoning of the oxidative enzymes within cells. In extreme cases, complete obstruction can cause anoxia, a total lack of oxygen, leading to rapid and severe cellular injury. Ischemia-reperfusion injury occurs when the restoration of blood flow and oxygen to previously ischemic tissues paradoxically causes additional cellular injury and death. This process involves several damaging mechanisms. Oxidative stress increases, leading to the production of free radicals. Intracellular calcium concentrations rise abnormally, disrupting cellular homeostasis. Inflammatory responses are triggered, and the complement system becomes activated, all contributing to further tissue damage beyond the initial ischemic event. This diagram illustrates the process of ischemia-reperfusion injury. In the upper image, an obstruction in the blood vessel, such as a thrombus, causes anoxia, leading to swelling and damage of the affected cells. In the lower image, the restoration of oxygen during reperfusion leads to the generation of reactive oxygen species, such as superoxide, hydrogen peroxide, and hydroxyl radicals. These highly reactive molecules further injure the already stressed cells, resulting in necrosis. Thus, while restoring blood flow is necessary, it can also trigger significant oxidative damage if not carefully managed. Free radicals and reactive oxygen species are major contributors to oxidative stress within the cell. A free radical is an electrically uncharged atom or group of atoms that contains an unpaired electron, making it highly reactive. These unstable molecules can damage cellular components by initiating lipid peroxidation, altering proteins, damaging DNA, and impairing mitochondrial function. If unchecked, the cumulative effects of free radical damage can lead to cell dysfunction and death. This diagram highlights the role of reactive oxygen species, or ROS, in cell injury. Under stress conditions such as radiation, toxins, or reperfusion, cells produce superoxide, hydrogen peroxide, and hydroxyl radicals. These reactive molecules cause significant cellular damage. Lipid peroxidation compromises the integrity of cell membranes. Protein modification can disrupt enzyme function and structural proteins. DNA damage can result in mutations, contributing to diseases like cancer. Although cells have antioxidant systems to neutralize ROS, excessive production can overwhelm these defenses, leading to widespread cellular injury. Chemical or toxic injury occurs when cells are exposed to harmful substances, known as xenobiotics. These agents can be toxic, mutagenic, or carcinogenic. Common examples include carbon tetrachloride, lead, carbon monoxide, ethanol, mercury, and a range of social or street drugs. These chemicals interfere with cellular metabolism, damage cell membranes, and can trigger mutations that contribute to disease progression, including cancer. Chemical agents, including medications, represent a significant cause of cellular injury. Both over-the-counter and prescribed drugs can be harmful if misused or taken in excess. Opioid abuse, in particular, has become a widespread public health crisis. Alarmingly, medications are now the leading cause of child poisoning, highlighting the need for greater safety measures and education around drug storage and usage. Environmental toxins are another major cause of cellular injury. Exposure to air pollution, both indoors and outdoors, can introduce harmful substances into the body, damaging respiratory and cardiovascular tissues. Heavy metals like lead, cadmium, arsenic, and mercury are particularly toxic, interfering with essential cellular processes and leading to widespread organ damage. Ethanol, commonly known as alcohol, can also cause significant harm. Prenatal exposure to ethanol can result in fetal alcohol syndrome or broader fetal alcohol spectrum disorders, leading to lifelong developmental and functional impairments. Unintentional and intentional injuries are a major cause of morbidity and mortality, occurring more frequently among men and showing higher rates among Black populations. Blunt force injuries result from the application of mechanical force to the body, leading to tearing, shearing, or crushing of tissues. These injuries are commonly seen in motor vehicle accidents and falls. Specific types of blunt force injuries include contusions, or bruises, lacerations, which are deep cuts or tears, and fractures, where bones are broken under force. Sharp force injuries result from the application of a sharp object to the body, causing distinct types of wounds. An incised wound is a clean cut that is typically longer than it is deep. A stab wound is deeper than it is long and often caused by knives or other pointed objects. Puncture wounds occur when a narrow object penetrates deeply into the tissue, while chopping wounds involve heavy, edged instruments like axes, combining sharp and blunt force. Gunshot wounds are another form of penetrating trauma and can vary greatly in severity depending on the weapon, distance, and location of injury. Asphyxial injuries occur when cells fail to receive or use oxygen properly, leading to cellular damage and death. Suffocation results from the lack of oxygen supply, often due to choking. Strangulation involves external pressure on the neck, seen in hanging, ligature strangulation, or manual strangulation. Chemical asphyxiants, such as carbon monoxide, cyanide, and hydrogen sulfide, interfere with oxygen delivery or utilization at the cellular level. Drowning is another form of asphyxial injury, where the airway is submerged in liquid, preventing oxygen intake and leading to hypoxia. Infectious injury is caused by the pathogenicity or disease-producing ability of a microorganism. The severity of the injury depends on the microorganism's ability to invade and destroy tissues, produce toxins that disrupt normal cell function, and trigger hypersensitivity reactions within the host. These mechanisms can lead to a wide range of diseases, from localized infections to widespread systemic illness. Immunologic and inflammatory injury occurs when substances generated during the body's inflammatory response inadvertently damage healthy tissues. Phagocytes, which are meant to destroy pathogens, can release harmful chemicals that injure surrounding cells. Biochemical substances such as histamine, antibodies, lymphokines, complement system products, and proteases also contribute to this process. Additionally, inflammation can cause alterations to the cell membrane, further impairing normal cellular function and promoting injury. One of the major manifestations of cellular injury is the accumulation or infiltration of various substances within the cell. Normal cellular substances, such as water, proteins, lipids, and carbohydrates, can accumulate when metabolic processes are disrupted. In addition, abnormal substances may also build up. These can be endogenous, produced within the body, or exogenous, originating from outside sources. These accumulations can impair cellular function and contribute to disease development if not properly managed. Cellular accumulations can arise through four primary mechanisms. First, there may be insufficient removal of normal substances due to problems with cellular transport systems. Second, cells might accumulate abnormal substances because of defects in their synthesis or metabolism. Third, inadequate metabolism of endogenous substances can occur when there is a lack of necessary lysosomal enzymes. Finally, harmful exogenous materials, such as pollutants or infectious agents, can infiltrate the cell and cause injury. Each of these mechanisms can disrupt cellular homeostasis and contribute to disease progression. This diagram visually explains how different mechanisms lead to cellular accumulations. Abnormal metabolism can result in the buildup of substances like fat in the liver. A lack of specific enzymes, as seen in lysosomal storage diseases, prevents the breakdown of complex molecules, leading to their accumulation. Protein mutations can cause defective folding or transport, resulting in the buildup of abnormal proteins inside the cell. Finally, the ingestion of indigestible materials, such as certain environmental particles, can overwhelm the cell's ability to clear them, leading to the accumulation of harmful exogenous substances. Cellular death is attributed to two primary mechanisms: necrosis and apoptosis. Necrosis involves the rapid loss of plasma membrane structure, swelling of organelles, mitochondrial dysfunction, and a breakdown of normal cellular architecture. Unlike apoptosis, necrosis does not follow a controlled sequence and often triggers inflammation. Interestingly, some forms of necrosis may still be regulated or programmed by the cell. Autolysis, or autodigestion, occurs when the cell's own enzymes break down cellular components after death. Coagulative necrosis is a type of cell death typically caused by ischemia or infarction. It is characterized by the denaturation of proteins, including albumin, which changes from a gelatinous, transparent state to a firm and opaque substance. Although the cells are dead, the basic tissue architecture is preserved for a short time, allowing for identification of the affected area. Coagulative necrosis is most commonly seen in organs like the heart, kidneys, and adrenal glands following a loss of blood supply. This image shows an example of coagulative necrosis. The tissue has retained its basic shape, but the cells within it are dead, and the affected area appears firm and opaque. The infarcted region demonstrates the classic features of coagulative necrosis, highlighting how ischemia leads to the preservation of structural outlines even as cellular detail is lost. Liquefactive necrosis primarily affects neurons and glial cells of the brain. During this process, cells are digested by their own hydrolytic enzymes, leading to the complete softening and liquefaction of tissues. Liquefactive necrosis is often triggered by bacterial infections, especially those caused by organisms such as Staphylococci, Streptococci, and Escherichia coli. Unlike coagulative necrosis, the structural framework of the tissue is lost, resulting in a fluid-filled cavity. This image demonstrates liquefactive necrosis in brain tissue. You can see how the normally firm structure has broken down into a soft, liquefied mass. The loss of cellular architecture is a hallmark of liquefactive necrosis, often associated with bacterial infections or severe ischemic injury to the brain. Caseous necrosis is most commonly associated with pulmonary tuberculosis infections. It represents a combination of both coagulative and liquefactive necrosis. The resulting tissue appears soft, granular, and cheese-like. Although the underlying cellular architecture is destroyed, the affected area maintains a distinctive appearance that is recognizable under the microscope. This image shows caseous necrosis, a distinct form of tissue death most commonly associated with pulmonary tuberculosis. It gets its name from the cheese-like appearance of the dead tissue, soft, white, and crumbly. Histologically, it represents a combination of coagulative and liquefactive necrosis, where the affected tissue loses its structure and is infiltrated by immune cells and cellular debris. This type of necrosis is typically surrounded by a granulomatous inflammatory response, as the body attempts to wall off the infection. Fatty necrosis most commonly occurs in the breast and other abdominal organs. It results from the action of lipases, which break down fat cells and release fatty acids. These fatty acids then combine with calcium and other elements to form soap-like substances, a process known as saponification. The affected tissue appears opaque, chalky white, and firm, providing a distinct visual cue during examination. This histological image displays fatty necrosis. You can observe clusters of necrotic fat cells, appearing as pale, opaque areas surrounded by an inflammatory response. The chalky white appearance results from the combination of fatty acids and calcium salts, forming soap-like deposits within the tissue. Fatty necrosis is commonly seen in cases involving trauma to fatty tissues or pancreatic injury. Gangrenous necrosis refers to the death of tissue due to severe hypoxic injury. It is commonly classified into two types, dry and wet. In dry gangrene, the skin becomes dry, shriveled, and takes on a brown or black coloration. In wet gangrene, the affected area becomes cold, swollen, and black, often accompanied by a foul odor. Wet gangrene is frequently complicated by bacterial infections, especially by organisms like Clostridium, leading to a more aggressive and life-threatening condition known as gas gangrene. Apoptosis is the process of programmed cellular death. Unlike necrosis, it is an active, energy-dependent process that is highly regulated. Apoptosis plays a critical role in maintaining tissue health by removing damaged, infected, or unnecessary cells without triggering inflammation. It can occur under both physiologic conditions, such as normal development and aging, as well as pathologic conditions, such as when cells are injured beyond repair. Autophagy is a self-destructive yet vital survival mechanism. During autophagy, cells deliver damaged cytoplasmic components to lysosomes for degradation and recycling. This process helps cells survive under conditions of stress, such as nutrient deprivation. However, autophagy also contributes to the aging process by gradually diminishing cellular function over time. Proper regulation of autophagy is essential for maintaining cellular health and longevity. As we examine aging and altered cellular and tissue biology, it is important to distinguish between aging, disease, and lifespan. Aging is a natural process distinct from disease, although it can contribute to disease vulnerability. Normal lifespan refers to the maximum potential length of life, while life expectancy describes the average years a person is expected to live. A key feature of aging is frailty, which involves weakness, decreased stamina, and functional decline in older adults. Frailty significantly increases vulnerability to falls, disability, disease, and death. Somatic death refers to the death of the entire body, marking the end of all biological functions. Following somatic death, post-mortem changes occur in a predictable and diffuse sequence. Pallor mortis is the paleness of the skin due to lack of blood circulation. Algor mortis describes the cooling of the body. Rigor mortis is the stiffening of muscles after death. Livor mortis refers to the pooling of blood in dependent parts of the body. As time progresses, putrefaction begins, leading to decomposition and ultimately skeletonization of the remains.